Okay, so in the last video, we have been dealing with the various types of the lipoproteins, which are the different types of lipoproteins based on the density or based on the speed, difference in the rate of the mobility. They can be divided into 5 different types: chylomicrons, VLDL, IDL, LDL, HDL. And of course, free fatty acid is also mentioned here, even though it is not a lipoprotein. Why free fatty acid is mentioned? Because fatty acids cannot be freely uh, 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 circulated in the plasma, and hence it will be always attached to another protein, but it is a albumin, right? It is not apoproteins. It is a pure albumin that is responsible for transporting the free fatty acids. So since free fatty acids are in conjugation with the albumin, we also include free fatty acids when we are talking about the lipoproteins. Okay, so free fatty acid is actually not a lipoprotein. It is free fatty acid which is in conjugation with albumin. So actually the lipoproteins are chylomicrons, VLDL, IDL, LDL and HDL. Right, since we can have when a serum sample, when a lipemic sample is centrifuged, we can have free fatty acid at the bottommost layer, right? So that is why free fatty acids are also mentioned along with the other lipoproteins, and all the other metabolisms of lipoproteins are over, and HDL we have started also. So HDL is the good cholesterol. It is otherwise known as good cholesterol, high density lipoprotein. And why it is known as good cholesterol? Because when compared to that of the other lipoproteins, HDL is having an apoprotein known as ApoA1. This ApoA1 is anti-atherogenic in nature, while LDL is atherogenic in nature, right? LDL is atherogenic, while also LPA, lipoprotein A, is also atherogenic in nature, right? Because they resembles plasminogens, and it impairs the fibrinolysis, so that clot formation is enhanced by lipoprotein. And in case of the LDL molecule, there is ApoB100. So this ApoB100 is highly atherogenic in nature. That's why LDL is known as the bad cholesterol. Lipoprotein A is known as the little rascal and all. While HDL is known as good cholesterol because it's having the apoprotein ApoA1. So what's the peculiarity of ApoA1? ApoA1 is the apoprotein, or it is the protein which is responsible for activating an enzyme known as lecithin cholesterol acyl transferase. So what's the function of this enzyme? We know that HDL, the metabolic function of HDL is to transport cholesterol or cholesterol esters from the peripheral tissues to the liver. So while LDL transports the cholesterol esters from the liver to the peripheral tissue, making a person fat, while HDL carries out the exact reverse phenomenon, in which the cholesterol esters are transported from the peripheral tissues to the liver. So how can it be made possible, or what is the relevance of this transportation? It is that this cholesterol is mainly it will be present in the free form. So this free cholesterol has to be first converted into the esterified form. That is, the cholesterol which is having a 3-hydroxyl group, or the hydroxyl group on the third position, will be esterified with a fatty acid. So when cholesterol is esterified with fatty acid, we call it as cholesterol esters. So any type of fatty acid can come and get attached onto the 3-hydroxyl group by displacing the hydrogen atom. Or the H is displaced, or it is substituted with a fatty acid. Then we call it as a esterified cholesterol or cholesterol esters. So what is the purpose of this esterification? So that it is the cholesterol esters, it is packed into the lipoproteins and can be transported easily, right? So this cholesterol esters, in case of HDL, the esterification is happening with the help of PUFA. It is not any other fatty acid which is coming and getting attached. The third hydroxyl group of the third position of the cholesterol molecule is esterified with the polyunsaturated fatty acids. Again, we know what are the beneficiary effects of polyunsaturated fatty acids, right? It protects the body, right? Again, the free radical scavenging, free radical attack and all. So it is, they can have the polyunsaturated fatty acids, since they are having number of double bonds, they are having the capacity to attack the free radicals or remove the free radicals whenever it is found. So these PUFAs, polyunsaturated fatty acid, a single molecule of polyunsaturated fatty acid will be coming and getting esterified onto the third hydroxyl group, the cholesterol molecule. So how can it be made possible? How can the PUFA comes and get attached onto the cholesterol? It is with the help of the enzyme LCAT, that is lecithin cholesterol acyl transferase. So from the name itself, what is the function of this enzyme? This enzyme carries out the transfer of the lecithin molecule or the polyunsaturated, the lecithin is transferring an acyl group from itself to the cholesterol molecule. That is lecithin cholesterol acyl transferase. An acyl group is being transferred from the lecithin molecule onto the cholesterol molecule, right? Onto the free cholesterol. So what is lecithin? In fact, lecithin is a phospholipid. It is phosphatidylcholine. So where is it present? It is present on the cell membranes. So the lecithin molecule or phosphatidylcholine which is present on the cell membrane is attacked by the LCAT, lecithin cholesterol acyl transferase, so that this lecithin is having polyunsaturated fatty acid in its structure, right? So lecithin is having polyunsaturated fatty acid on its second carbon atom. So the second carbon atom, the PUFA from the second carbon atom is cut. It is removed from the lecithin molecule and it is translocated from the lecithin onto the third position or third hydroxyl, the cholesterol molecule. So that free cholesterol is now converted into a protective form, that is the cholesterol esters. While lecithin is converted into lysolecithin, that is the one of the fatty acid among the three carbon atoms is replaced with a an OH group or an H hydrogen atom come from the cholesterol and it's getting located. It will be getting fixed onto the second carbon atom of the lecithin, and in exchange, what does the lecithin do? The polyunsaturated fatty acid which was on the second carbon atom is donated to the cholesterol molecule and it is getting fixed onto the OH group. H is removed and onto the position of this H, the PUFA will be coming and getting attached, right? So what is the role of HDL in this reaction? It is the ApoA1 that is present on the HDL molecule is responsible for activating the LCAT. The enzyme, only when the enzyme catalyzes the reaction, then only the cholesterol, free cholesterol will be converted into cholesterol ester, right? So lecithin is converted into lysolecithin and the free cholesterol is converted into cholesterol esters only in the presence of the enzyme LCAT. And this LCAT enzyme is subjected for activation by ApoA1. So that is why ApoA1 is anti-atherogenic in nature and which makes the HDL a good cholesterol. Okay, so again, another important factor is that it is a rich donor of ApoE and ApoC. Whenever VLDL, IDL, LDL, etc. is in circulation, these molecules are being supplied with more and more quantities of ApoE and ApoC, and they are all derived from the HDL which is circulating in the plasma. So they are acting as a rich reservoir of ApoC and ApoE, which will be donated to the VLDL, IDL, etc. Then carrying out the lipoprotein cascade pathway, right? Then they are delivering cholesterol esters to the liver for the uptake by the hepatocytes. So the protein which is responsible for this is the SRB1, that is reverse cholesterol. This cholesterol which is being transported from the peripheral tissues to the liver is known as reverse cholesterol transport, right? And it is having protective effect. It is anti-inflammatory in nature. It is antioxidative. The platelet anti-aggregatory, that is platelet aggregation is prevented. That is anticoagulant, pro-fibrinolytic activity, that is it prevents the clot formation, etc. So in effect, HDL is highly protective in nature. Right, so this is a overall metabolism of all the lipoproteins. That is, liver is there, intestine is there, right? So the from the intestine, the dietary lipids are taken up by the chylomicron molecule, which is having ApoB48 on it. The ApoB48 is synthesized by the intestine, right? This chylomicron will be converted into chylomicron remnants by the continuous release of TG and cholesterol esters to the adipose tissue and skeletal muscle, so that finally the size will be reduced to give rise to chylomicron remnant. And during the course of this conversion, what is happening? ApoC and ApoE will be continuously supplied to the chylomicron molecules by the HDL, which is present in the plasma. Right now, this chylomicron remnant will be delivering the ApoC back to the HDL, and ApoE will be accepted by the chylomicrons and all, and they finally reaches the liver. Now, regarding the other apoproteins, the liver, which is so that is how the dietary lipids are reaching the liver with the help of chylomicrons. And once its half-life is attained, it will be subject for catabolism, or it will be subject for breakdown. It is happening in the liver and all. And in the liver, another proteins, that is VLDL, is synthesized. It is another lipoprotein whose function is to carry out the transport of the endogenous TG. and cholesterol, right? Mainly the TG. So the VLDL is having the apoprotein ApoB100. So even though both the ApoB100 and ApoB48 are products of the same gene, ApoB100 is synthesized in the liver while ApoB48 is synthesized by the intestinal cells or enterocytes. Right? So the gene is same, but this gene in the intestine will be undergoing a modification which is known as mRNA splicing, so that the size is reduced to 48% of the total size of the ApoB100. Right? So VLDL, that's the only difference between chylomicron and VLDL. Now, VLDL functions almost the same. One transports exogenous TG while the other transports the endogenous TG. So, VLDL transports the endogenous TG and is released into circulation. And again, during the course of the journey, what is happening? ApoC and ApoE gets deposited, which is derived from the HDL in the plasma. Right now, VLDL will be reducing its size to give rise to VLDL remnants, followed by IDL with the continuous release of TG onto the adipose tissue, other tissues, peripheral tissues and all. Right now, VLDL will be converted to IDL, and IDL will be finally converted to LDL with the removal of all the other apoproteins, and the ApoB100 will be only present when LDL molecule is formed. All the other apoproteins will be removed. Right. So this is the overall metabolism. Now, metabolism of HDL. HDL is having, it is responsible for transporting the cholesterol LDL. What does the LDL do? So the LDL molecule thus formed. Majority of the LDL will be formed from VLDL, and it is responsible for, once a VLDL is converted into LDL, it will be almost devoid of TG. It won't contain any TG. TG will be present only in trace amounts. All the remaining TG will be supplied to the peripheral tissues. So such a molecule we call it as a low density lipoprotein, because LDL molecule contains only the cholesterol esters. Only trace amounts of TG can be found in the LDL molecule. All the remaining TG will be removed. As a result of this, what happens? The density is getting increased, right? And what does this LDL do? LDL now start delivering the cholesterol esters, which has been derived from the liver, because from the liver VLDL is formed. VLDL contains majority TG and cholesterol esters, and TG will be gradually getting decreased, converting VLDL into finally LDL. Now the LDL is having only cholesterol esters. These cholesterol esters will be now supplied to the peripheral tissues, right? So the function of the LDL is to transport the cholesterol from the liver to the peripheral tissues. Now the HDL carries out the reverse cholesterol transport. That is, it transports the cholesterol or cholesterol esters which are present on the peripheral tissues, present in the peripheral tissues, and they will be taken into the liver. Right, so their HDL molecules are synthesized in the liver and also in the small intestine, and we having apoproteins also, mainly the ApoA1, and it is also rich in ApoEH, ApoC. Right, and when secreted, they contain little cholesterol or no cholesterol esters, and gradually what happens, once it is synthesized in the liver or in this intestine, it is not having any cholesterol esters. But as they move towards the peripheral tissues, and once they reach the peripheral tissues and gets attached onto the peripheral tissues with the help of the apoproteins, they enters into the peripheral tissues and collects the cholesterol esters from there, and they will be loaded with what large amounts of cholesterol esters, so that the nascent HDL, which was initially discoid in shape, disc shaped, like oval shaped in an HDL, when they are laden with cholesterol, what happens? The shape will be converted into a sphere. Right, that's known as the mature HDL, or they will be having, this is known as HDL. It is known as HDL2. That is HDL, the mature HDL. Right now, this mature HDL now comes out of these peripheral tissues, and they start moving through the circulation. And during the course of the circulation, what happens? They will be continuously liberating the ApoC, ApoE, and the cholesterol esters will be given or will be transferred to the other lipoproteins that they come across. When this transport is given, or this transport is carried out by cholesterol ester transfer protein, CETP. Right now, so that as they move towards the liver, what happens? Slowly the cholesterol content will be getting reduced, and this cholesterol, when they are giving the cholesterol esters to the VLDL, LDL, IDL, etc. In return, what happens? HDL accepts the TG, right? So that in return, they will be accepting the TG from those lipoproteins. That's VLDL. VLDL is responsible for transporting the endogenous TG, right? So this endogenous TG containing VLDL will be slowly delivering the TG, right? Triacylglycerol to the peripheral tissues, to the adipose tissue, skeletal